this works. Welcome everyone to this panel session. The title this afternoon is The Future of Genetic Medicine. My name is Nina Bauer. I'm the Chief Business Officer of SmartCella. We're a small but very well-established biotech company based in Stockholm, Sweden, with both cell therapy technologies and therapies in development, as well as delivery technologies. So welcome to this afternoon's session and for staying with us. I would like to ask the panel members to introduce themselves briefly and also maybe give a quick statement as to what you think is the future of genetic medicine. Behnam?
Yes, thank you. And first of all, thanks for the ARM, for the opportunity to be here. And my name is Behnam Baghbaderani. I'm heading the commercial for Lonza Cell and Gene Technologies. And as you might know, Lonza is a CDMO who has been active in this space for 14, 15 years, essentially serving our customers who are interested in cell and gene therapies and taking their cell and gene therapies from clinical to commercial. And this is important mission for us from the perspective of enabling and commercializing cell and gene therapies. And that's really my interest in this panel discussion, how we can make this cell and gene therapies happen and make it accessible to the patients in the future, essentially make the cell and gene therapies more affordable and accessible to a wide range of patients. And that's the discussion that we have today.
Thank you. Lisa?
Hi, everyone. Lisa Deschamps. I'm the CEO of a private clinical stage gene therapy company called AviadoBio. We are very much focused in CNS and drug and delivery overall. For me, the future of genetic medicine, very similar, is just making sure that we can actually bring more of these truly transformative and potentially curative medicines into the hands of as many patients as possible. I think we've had quite a number of successes over the last several years, but we really need to do better. And so hopefully in the future, we can really just continue to see that grow across many therapeutic areas and going from monogenic to even greater disease. So thank you.
Thank you, Lisa. Kinnari?
Kinnari Patel, President, Chief Operating Officer, and Head of R&D for Rocket Pharmaceuticals. For me, the future really means DNA is not your destiny. That sounds simple. But if you have a genetic disease, hopefully there's a gene therapy available to treat it. And that way we can actually change humanity. So that's my future vision. It's quite a lofty one, but that's the aspiration.
David, please.
Yeah, hi, so my name's David Venables. I'm CEO of a relatively new startup in the cell therapy space called Laverock Therapeutics. My journey with cell and gene therapy actually started back in the 90s, which makes me feel a lot older than I feel or think that I am. So it's always a shock. So I've been involved over that time with many sort of iterations of gene therapy companies. And particularly more recently thinking much more about gene regulation, how to control genes through the work that I did with forming and leading Symptomics. And now in Laverock Therapeutics, we're taking a totally new approach to gene silencing and creating programmable gene therapies. But I suppose looking back over 25 years of involvement in this field, I don't know if I'll be here in 25 years' time. I do dangerous things that probably mean that I won't. What should gene therapy and cell therapy look like in 25 years' time? Well, it should be just an accepted norm. It should be an accepted norm that you get diagnosed with a genetic disease, you can go and get there's meaningful treatments, and not just treatments that improve quality of life, but actually cure disease. And it's accepted and it's standardized and it's done in a cost-effective way that just makes this another normal modality. And that would be the dream. If we can get there, that would be perfect.
I think we're all aligned. And Richard, please.
Hi, everyone. My name is Richard Wilson. I am the SVP and primary focus lead for genetic regulation at Astellas, where I'm responsible for our portfolio of AAV gene therapy programs across neuromuscular, CNS, some ophthalmology, and cardiac areas. Boy, it's hard to be the last person to answer the question, right?
Sorry.
I would say all of the above. To me, it's a future state where all these amazing, powerful tools and technologies we're working on today are just really accepted. And I think I'll go one step beyond and say, even in non-genetic diseases, I think that this will just be normal, that we will be very accustomed to how gene therapies are applied to a range of diseases, how they're approved, how they're paid for, and how everyone who needs one can get access.
Thank you. And I would also like to encourage the audience to participate. So if you have any questions or want to contribute and challenge us a little bit, then please feel free to do so. Being here this week, actually, I've been talking to a good range of people, and it feels like we're at the cusp of something new. And when I say that, what I mean is that I think we're all aligned, and you've heard from my esteemed colleagues here, that we know that cell therapies work. We know that gene therapies work. We've seen it, they're approved, they're out there, people get treated every day these days. So it feels like we're on the cusp of, now that we know that it works, we need to make it better. We need to find better ways, more efficient ways to lower costs. We don't want to talk too much about costs today, but be more future vision. But it really feels like now we need to kind of almost revisit what we have been doing and find better ways and maybe re-engineer and design the Gen 2 of what we've been doing. And so I would like to ask my panel members here and how they envision that could happen from let's start with the product itself. How could that happen? Where are your individual companies working on that Gen 2? David?
Okay, so Gen 2. So what we're focused on in Gen 2 is programmable cell therapy. So this is the idea that you can develop a cell therapy that you administer to a patient, and that cell therapy then responds to the patient's condition, responds to the disease, and is adaptive in its therapeutic approach. And the way that the industry has looked at many of these things like this around gene regulation is to try and create sort of expression cassettes with all of the sort of synthetic biology principles of what a cassette needs to look like to perform, and then try and engineer and deliver that into the cell and hope that that cassette then performs in vivo in the way that it always performs great in vitro. Let's admit it, in vitro is the easy part. Put it into the complexity of a 3D human being or animal model, and everything starts to go wrong and fight against you. So the approach that we're doing is trying to take a totally alternative approach, not to create these cassettes and try and drop them into a cell, but actually look at the way that the cell already does an excellent job orchestrating its own gene function and its own timing of what genes get expressed or silenced, when and where, and in what response to what. And a lot of this is done through the transient expression of microRNAs. So we're essentially going in and we're editing endogenously expressed microRNAs that are naturally expressed in a particular condition, in response to a cell state, in response to a disease. And we're editing those and essentially converting them now into an endogenously expressed siRNA that silences our target gene under the same specific set of conditions that a natural microRNA would have been expressed in. So in that way, we can engineer and create a programmable cell that will continue to respond and adapt to the disease environment in vivo. So that's sort of thinking about a future generation of an advanced cell therapy that I'm really excited about.
And I think we've been seeing more and more talks this week about larger indications, and that obviously means we need to be more programmable and potentially have off switches. I don't know, Richard, I think you mentioned to me at some point that Astellas is working on some similar things in the viral vector space.
Yeah, no, we've got a couple of things that are kind of nearer term, I'd say. One is we're heavily invested in next-gen capsid approaches. We see just a ripening of the field right now, where going from kind of the natural serotypes that we're all familiar with and still have a lot of life in them, we're able to see various engineering approaches come up with capsids that both target organs more specifically, which has the benefit of not exposing organs that don't have a therapeutic purpose for gene therapy to target. It means you can also deliver a lower total dose, which is always a good thing. That means that your manufacturing runs are going to have more doses per batch, which helps us as we think about moving into larger patient populations and also helps address cost. The other thing I might highlight is our relationship we have with Taysha Gene Therapies, where they're applying a really interesting approach for genetic regulation, where they built in some microRNA recognition elements into their Rett syndrome cassette, where, for those who may be familiar with Rett syndrome, it's a mosaic condition where an individual patient has some healthy cells and some dysfunctional cells, and if you over or underexpress the protein of interest, MECP2, you see either Rett syndrome or MECP2 duplication syndrome, both of which you do not want to have. And so they've engineered in these regulatory elements which will actually recognize if the environment that gene therapy finds itself in has the required RNA signal, which will then in turn shut down expression. So, that helps to say we worry a lot about delivery. We worry a lot about concentration of gene therapies in different tissues. But if you can build in to the cassette itself a regulatory element that allows you to more accurately predict the therapeutic level of protein you need to produce, that's a really interesting approach for a range of diseases.
Thank you very much. And our company works on a catheter, so I think we can deliver straight into tumors and organs. We can help with that on top of it. But I think, Lisa, you also mentioned that you're working on delivery technologies. So anything?
Thank you. Yeah, so we're very much focused on, again, taking therapeutics that we know a lot about, like AAV9 and microRNA, and coupling them with novel direct delivery approaches. We're not doing novel capsid engineering at the moment, although we know lots of you are, and so we're looking forward to those deliverables that we can potentially leverage in the future. But we're just also thinking about, okay, what do we have today based on translation and clinical data where we can get to patients faster and get to broad groups of patients faster. And so, we kind of think of delivery in two areas. The first one is on the constructs themselves and making sure they can be targeted, efficient, and in our case, neuronal specific where they can be. And then the second area is around these direct delivery approaches. How can we go right into the target for that particular disease area? So in our lead program in FTD, which is frontal temporal dementia, where the disease starts out very much in the frontal lobe, but as it progresses, becomes global in nature. How do we use parts of the brain itself, so the thalamus, which is in the deep brain structure, to actually do the work with us? If we target the thalamus directly, we could use very, very micro doses, almost, of vector to deliver to the thalamus. The thalamus is kind of the sensory relay hub to the brain, and it is a secreted protein, so when you deliver it there, what you can see is really nice broad biodistribution across all the cortical regions of the brain. So again, just thinking innovatively, how do you take something you know a lot about, couple it with something novel, allow your own body, in a sense, to do the work for you and be able to hopefully translate to a maximal therapeutic benefit for patients while minimizing any safety aspects that we've seen in other gene therapies, where if you deliver it systemically, you have a lot of peripheral exposure in the liver, blood, et cetera. And so we're doing a lot of that work in our lead program, and then in our pipeline, we're very much focused also, same context, how do we get into the spinal cord for diseases that have much more spinal cord involvement? And there we have a proprietary, exclusive to us, delivery platform that we've licensed in, that we've taken on development for. So those are two areas that we're also working on.
Thank you very much. I want to ask a little bit of a provocative, maybe a wildcard question, so bear with me. Because David, you mentioned synthetic biology earlier, and actually incidentally at lunch today I was having a conversation with someone. This Gen 2, do we maybe need to completely rethink and start from a synthetic biology approach and take all the things that we need for cell and gene therapy, but build them from the beginning and leave out all of the stuff that is causing issues and complications and manufacturing problems and all of that? I don't know, do I have any volunteers on that topic? Go ahead.
Oh, I'll go for it. I don't mind controversy. As said to Nina, you hear too many panels where everyone's saying the same thing, so we need to try and stir things up a bit. So I have my interest in synthetic biology has just grown and grown. And so, I love viruses. They're great. I've been playing around with viruses, it feels like, all my life, either for viral vaccines or for gene therapy, or the fact that my kids, when they were young, were like viral production factories of their own and always infecting us. But the more and more I've got into this whole thinking and mindset, as molecular biology and as the tools and the capabilities have grown and developed, to see DNA, RNA, and cells as vehicles for coding is actually, I find fascinating. And when we were in Synpromics, 14 years ago, we came out with platforms to create synthetic promoters, which was totally unique. I mean, it's great now to hear so many companies talking about their synthetic promoter pathways, pipelines, et cetera, which is, that field has really moved forward. But I always had this feeling, wouldn't it be great if you could do amazing biology without the inefficiencies of biological systems? And I was always desperate to find people, particularly on the delivery side. I felt that we could engineer molecular circuitry. Senti, for example, came up with molecular circuitry. Now in Laverock, we're recreating synthetic circuitry with synthetic promoters. We had the ability and the tools to create synthetic circuitry. But at the end of the day, it always then started to fall over when it had to then be put into a biological system. And there's nothing worse, to be honest, as I say, I love viruses, but there's nothing worse than having to be plagued by having to rely on viruses. And so for delivery systems, I mean, it's great talking to people at this conference, the number of delivery LNP or vesicles or other deliverable modalities that are starting to come out. I was desperate for that 10 years ago.
You were ahead of the curve.
Design out of that and away from it. Now it's starting to come forward. So I just say to all of those guys looking at non-viral delivery systems, keep plugging away at it, keep working at it, try and get it right, try and get the targeting right, the specificity right, the dosing right, getting into specific cells, the ability to redose, because I cannot see a future where the mass production of gene therapy products for big indications is going to be achievable through a viral platform. That's my controversial statement. I would say challenge at me on that. Is virus really the future of large-scale treatment of indications? I don't see, for many reasons, why it can be, and we need alternatives that are more synthetic biology-based.
Well, yesterday, a company went live called Envelop, so they, I think, are trying to do exactly what you're saying. They're all ex-Bluebird, or at least the two founders are ex-Bluebird. I don't know, Kinnari, any thoughts?
I was going to say, I'll debate you on the viral component, right? When I first started in industry or drug development 20 plus years ago, biologics was a new cool thing, right? Then they were like, hey, if the viruses could be more manufactured, et cetera, right? I feel that while we have few therapies that have succeeded, let's be honest, it's been about a dozen therapies that have actually seen the day of light, right? So if we really want to focus on getting treatments for patients that really need it, advancing science, to me, if we talk about the future, not 150 years from now, but if I talk about 10, 20, 30 years from now, viral backbones to me seem like a feasible way to go after so many monogenic diseases and beyond, that figuring out and focusing on how to make that scalable, cost-effective, and even the dozen or so therapies, 13 therapies that have been approved, they're only accessible for those that are in Europe or US or have financial means to make it accessible. Yeah. But these diseases impact everybody across the world. So how do we use the viral backbone to make it cheaper, faster, better? To me, that is also innovation. It's a different, I know I'm at a cell and gene therapy conference where they'll say, hey, the true innovation is on platform science. To me, innovation is in every aspect of science, up to getting treatments to patients. So to me, innovation in drug development and platform approaches in making viral therapies more successful, accessible, and cost-effective, it's kind of the near future, 20, 30-year future that I see.
So can I keep digging my own hole?
Yes.
I don't know. I don't know. I mean, we have a big panel here. Maybe somebody else wants to contribute.
I wanted to maybe build on Kinnari's points as well as the case for viruses. Maybe a yes and, because I do think we need to avoid this kind of dichotomous thinking of it's going to be editing, no, it's going to be transfer, it's going to be LNP, it's going to be yes, it's going to be all of these things. I think two things really come to mind for me. One, from the last 2 days, is this sense that we're at a moment in time where we really need to start delivering to patients. We need to be able to solve access. There's people who are waiting for us. And as if you had to start from a completely blank slate, fine. But I think we have like an obligation as a sector to really follow through on the technologies that candidly, we've shown have worked. So I think demonstrating these products can deliver what they say on the label is important. The other point which strikes me is we've it's not a very sexy example, but we've demonstrated we can do viruses at scale. Anyone remember what Johnson & Johnson AstraZeneca did during the COVID pandemic? We used viral vectors to deliver vaccines en masse. Now, that's a much lower dose than we're accustomed to. But that to me is saying the idea of using a virus as a platform to deliver a therapeutic to a larger population isn't completely crazy.
I just wanted to elaborate on this topic, as I think there is nothing wrong with looking for futuristic, innovative approaches when it comes to delivery, synthetic, non-synthetic. But there is significant experience and there is significant expectation to deliver to the patient, as you mentioned. And I think from a CDMO perspective, in particular, process, practical aspects of the design and delivery, there are some significant considerations that would be worth to highlight. When it comes to the delivery, the importance of capsid design, GOI design, and then essentially, how can we improve efficiency, productivity, target delivery to the specific type of tissue. And unfortunately, there isn't much experience that is shared across the industry because of the competitive nature, because of IP, because of many other communication challenges. But there are also approaches that can be taken, particularly from a CDMO perspective. You can look at the design of experiments. You can look at the type of capsid design you have. We know that for AAV capsid, for example, the type of sequencing would impact the assembly, capsid assembly, the GOI packaging as well. There are significant impact on the productivity that is arise from the type of the GOI design. So there are particular ways of looking at improvements that are needed for the field, but obviously there are new innovative approaches that can be used. I think all together we need to look at this in perspective and what can be done to deliver the therapies to the patients.
You're looking like you have something to say, David.
No, I think, no, so, I mean, your point is absolutely taken. We are where we are today, and there is patient need, and there is a requirement, and there is an application, and there is value, and we should not stop what we're doing. But I think if this is talking about the future, where are we going in terms of this industry? My sense is that the viral modality has a ceiling that will be incredibly difficult to break through from a mass production perspective, from a cost perspective. Bear in mind that the safety profile of many of the diseases we're going to be focused on in future will be very different. And so viruses will have an inherent different safety profile in comparison to some other technologies. And so I think if we're going to sort of be realistic in forecasting where we need to get to, where do we need to apply our innovation and our energy, I think we need to start to seriously think in applying some real firepower behind non-viral technologies. And I say that from, I mean, part of my life I've spent as a vaccine developer, so I've taken products through FDA approval for mass manufacture of vaccines. There we were producing vaccines at sort of tens of euros a dose in a pre-filled syringe. Now that's a cost of goods and a scale of operation for a vaccine, which is, I can't see AAV gene therapy getting there. For many years, I'm just not seeing the pace of innovation that we need to actually get there. And you go back to my other life, which was in the early days of monoclonals, when Celltech Biologics, Lonza, were innovating in monoclonal antibody production. I say this because a lot of people say, well, we did it with monoclonals, so we're going to do it with viral vectors. I don't think that model, that monoclonal model, translates into something that is directly applicable to what we see in terms of the dynamics and the structure of an AAV manufacturing process, for example.
But I do think I think we're all in agreement. It's not an or, it's an and, right? I think we should look at innovation very broadly, to your point. We should do exactly what you're saying, and I think also to the points that Kinnari and Richard mentioned, right? We should think also about the things that we have today in our hands and how can we do them more innovatively, right? Whether that's more efficiently from a cost perspective, whether that's, more, how can we also get the same achievement using very little in different delivery modalities. I think there's so many ways we can look at it, and I don't think it's an or, I think it's an and, right? But I think what we all agree with, is my guess anyway, is that we don't want to stop in the meantime, right? We want to keep delivering to patients across today as well as tomorrow.
I think we have consensus that the developers right now need to continue optimizing, and then the innovators out there need to do everything they can to get us to the next step. Okay? Good.
Anyone from the audience want to object? I'll take that.
Yeah, any objections to this in the audience?
Purposely trying to poke the bear on this one, to be honest, so responding.
Agreeing, we're agreeing.
No objections? But you guys touched on manufacturing optimization. We haven't talked about that yet. Obviously, that's a broad and wide field. In terms of, we touched on when we spoke earlier a bit on like platforms and how can we optimize and be more efficient by having approaches that are a little bit more plug and play, and then also in terms of approval pathways, be more efficient. Do you want to start, Behnam?
I can start. So I think it is clear that platform technologies are needed in able to make the manufacturing processes more robust and also reduce the cost down. It's also interesting to see that even the regulatory agencies, in one of the most recent presentations that I saw, they are also looking at vending machine type of approach for the cell and gene therapies and using platform technologies. So everybody is looking at how we can enable platform technologies. Some key considerations when it comes to the platforms. The type of technology is important and the choice of the platform, whether we are there from technology development perspective, scalability, and then the ability to move and apply, moving from one GUI to another GUI, robustness of the platform, and it will be also somewhat specific to the indication and application. There will be significant development need when it comes to the platform itself, but then once we have a platform, whether it's plug and play, it will come down to level of characterization, process product characterization that has been done using certain type of platforms.
And I can add to that a little bit. So I've been, for the last 2+ years, been talking to Dr. Peter Marks and Dr. P.J. Brooks at NIH, right, on platform approaches. You can think of a platform in multiple ways, right? So at Rocket, we have the benefit of 6 programs and a lot of data collected, from plasmid stability to vector to everything you could imagine. So as I look at this data, and if we think about it, what do we need to rinse and repeat 10+ years of drug development, stability, endpoints, analytics, in-process testing, final testing for every single product, every single batch, right? Because that is what's adding to the cost of goods and the actual product development costs, right? So if I look at Fanconi anemia, Rocket has submitted an MAA and soon will submit a BLA in the US for Fanconi anemia A subtype. So that 70% of Fanconi anemia population could be served with this. Well, Fanc C and G patients have reached out saying, what about us? So do I need to do preclinical all over again from mice, monkeys, all the way to humans? Or can we actually talk about, hey, if all I'm doing is on one of the plasmids, changing a transgene, right? Disease is the same, the endpoints are the same, the patient population is essentially the same, the manufacturing is the same CDMOs, right, with Lonza. So if everything's the same and you're only changing 1 or 2 things, why do you have to do all of it all over again? No. Because these patients they're willing to take the risk and the benefit risk. If you can we do synergy in preclinical models? Can we do synergy in the clinical? Instead of having a 15-patient phase 1, phase 2, can I do a 4-patient study for C and maybe 4-patient for G to expand that market access to patients, right? So to me, platform approaches can be applied to scaling up. Whether you go from R74 to AAV9 is another way to think about it, or you go to 800 liter to 2,000 liters, but it could also just mean different disease gene and transgene being changed around.
No. I was talking to Jen Moody about something similar from Danaher, because she's in charge of the Beacon projects, and the one that she has just signed with Jennifer Doudna on the CRISPR side, they're we're talking about access for bigger indications, but equally, a lot of the orphan and ultra-rare diseases need that kind of approach, because what they're trying to do is develop platform manufacturing, and then only the GOI changes. And so they're having exactly the same thoughts on everything else is the same. Can we do basket trials or whatever you want to call it and make it a lot more efficient? Any other thoughts on that?
Yeah, no, I think we're doing something very similar in ALS, very much similar to what was just said, where we're looking at, efficiency, operational efficiencies, obviously as benefits for patients most importantly, but industry, regulators, et cetera. So we think we take our microRNA platform, we can just exchange out, plug and play the different targets, leverage the data, good use ethically for sensible use of animals, right? So you're not, doing all this additional preclinical work in vivo that you don't need to do, and at the same time, ultimately bringing these therapies much faster to the market. And from a regulator perspective, we're doing, we're leveraging the umbrella, kind of trial and doing master protocols for different products, but same disease to the points mentioned. So again, ultimately, we can treat more broadly the disease, more patients, hopefully, and again, really expedite the timelines from, discovery to commercialization for patients. So lots of utility.
I wanted to highlight two aspects. Again, I'm going back to my earlier technical life at Lonza and industry. When it comes to platform, I think there are two key considerations. One is robustness of the platform, which comes from the level of analyticals that are included in the platform, because you would want to essentially be able to plug and play and apply from one GUI to another GUI, from one target to another target, and there you need to have a robust analytical methods included. And then also having some characterization methods to be able to gather and collect the data. So data is becoming very important when it comes to understanding and learning from one platform as you move from one delivery to another one. I think these are the two considerations that I would highlight as a CDMO for the parties out there.
Maybe just to add on that point, is I feel as though we sometimes underestimate how far we've come, and that we have tools today that measure and can characterize products that we can see things we didn't even know were a thing like 4 years ago, right? And part of the challenge we've had is, processes that were developed way back when now look a lot more wrinkly than we thought they did at the time. But now, as those analytics and the tools are maturing, we're able to say, all right, now we can establish what that baseline should look like. I think as well, there's a lot of optimization that's going on. I think this is a below the waterline type topic, where I know within Astellas, we've done a lot of work looking at next-gen processes that we're starting to try and bring online. I know other companies we talk to are also having similar things, similar thoughts. It's not being presented yet, but I do think, we're accustomed to this, the cost boogeyman of being like, oh my goodness, it's 6 figures a patient. I think 4 figures a patient is going to become normal pretty soon. And it's just, probably 60% of the people in this room are nodding along quietly, but would be shot if they said so out loud by their IP attorneys.
But actually, I want to see, are there any questions? We really would like some questions. No? Yes, Francesca.
Be careful what you wish.
No, she's friendly. We're good.
We didn't plant that.
I'll try.
Is the box on? Yes. Yeah. Actually, I have a question for Kinnari, and you were saying...
oh, I just said you were friendly. We know you.
Oh,
okay,
yes.
Be nice.
Well,
I said be careful
question.
for what you wish.
You teed us up. You said you've been talking to the NIH and talking to Peter Marks. What are they telling you in response to your rinse and repeat? What are they telling you to work on? How is that being approached? Are they saying you're still dreaming a pipe dream, or... What's the response?
Here's the good thing. In 3 years, what I've seen in advancement of the same questions that I'm asking, theoretically, we're all aligned. So I think that's the good news. So NIH, FDA, EMA, we all want to get there. I think practically speaking, how do you apply that per each disease, per each platform? I think that's a conversation that's quite challenging for FDA to tackle, right? So if you're thinking about it from FANC A to Fang C or G, I think it's simpler for the FDA to get there or the health authorities to get there. But if you're thinking about it from a different perspective, I think it all comes down to analytics and data. How robust is your data package, and how much can you tell what the differences are in that platform, and how do you control for that or differentiate for that? And I think that's where the rubber hits the road, right? It's going to take few companies like ours having these conversations informally, formally, by programs like Fanconi anemia to figure out, is this reality that's going to happen in the next few years, or are we going to wait for guidance 5, 10 years down the road to make it a reality? So to me, I think the good news is the interest from senior management, both at FDA and EMA, is there, absolutely. I think the other thing that I've seen, which I love in the last 18 months, is EM and FDA are, in the cell and gene arena specifically, they're having monthly conversations on topics like platform, on topics that are on late-stage assets. So I think that synergy will help us get there. So I think we just need to do more conversations, more concrete examples. So I do advocate for those of you that are sitting and listening to this, if you have a program that could apply, go test it out. What's the worst thing you're going to get? No, you have to go back to preclinical, clinical, right? But until we test out a few of these examples that are more so black and white than in the gray zone, I think the more we realize what's feasible. So I'm excited. I'm actually talking on a panel next week with Dr. Peter Marks about this exact topic. So.
Yes, please.
Hey, so I guess directed at David a bit, but anyone can answer. As you know, I'm a huge fan of the mix of emergent synthetic biology and genetic medicine, and a huge believer in the need to add control logic to these things. What do you think is going to be the first one to really break that ground open? I mean, I've been watching preclinical development on this for almost a decade. But when do we start seeing these, and what do you think the objections are from the regulators about trying to implement some of them, or limited it so far?
I missed the key point. Implementing regulator. I missed the key.
A little hard to hear for us. So sorry.
Are these going to become more integrated into therapies? What's really holding them back right now, and what do you think or what have you heard the regulators saying about attempting to implement control logic like this into these payloads?
From a synthetic biology perspective?
Yeah.
Okay, so
control logic.
I'm trying to paraphrase. It's really hard to hear. When are we going to see the synthetic biology technologies get to the clinic? Like, what is your prediction on that?
So they're already in the clinic. I don't know how much I can say, but somebody else could, because of the confidentiality. But I know promoter constructs that were created through the Synpromic synthetic biology platform are now in the clinic, and there's maybe a company in the room that would be happy to talk about that. So I would say they are, and there are other constructs that are in the clinic. So it's happening. And this is the whole thing about trying to make it normal and not try to make it special. Just make it a sort of a language, a mindset, an approach that just is logical, and it solves a problem, and it gets better solutions to patients. And I think if we shouldn't sort of, and maybe I'm a danger of sort of labeling synthetic biology as how excited I am about it, because it then becomes a thing. It shouldn't be a thing. It's a process and an approach to think about what is the best design of a product that's going to have most impact on the patient. And those designs and those design principles are working through development pipelines, preclinical development, and they are now in the clinic. Yeah, it's happening.
Maybe one thing to add, I agree completely. I think these kinds of products are heading into the clinic. I think obviously specific feedback is going to be impossible to share for almost every sponsor, but I think there are probably some trends we could think about, and that's going to be about translation. And I think every regulator is going to want to know, how do I know that this fancy circuitry is going to actually be safe in humans? Fantastic, you've solved the problem for mice, but what's my confidence that that's going to translate? And I think that's going to push the challenge back to saying, where are I with, getting smarter about using organoids, other systems that maybe don't rely on having to jump species, to say to a regulator, are you ready to roll a dice on this? Because, we believe that this non-clinical package is robust. So I think maybe it's that kind of seeing more of those thoughtful preclinical designs will be important.
Thank you. Any more questions? If not, I wanted to touch on something. I think it was you, Richard, who was touching on data generation and testing and analytics, and that we're going back to all the things that we have done 10, 15 years ago, and we're now understanding better. But I think the other question is, where does quality stop as well? Because I think the more we test, the more we figure out, the more answers we get to questions that we never asked. So I don't know, anyone on that one in terms of quality, since we're optimizing everything right now?
So I mean, I have a first-hand experience of back to my sort of viral vaccine days, where the cost of goods of a batch, over half of it was analytical testing costs. And obviously the biggest reason for batch failure was levels of imprecision and control around analytics. So we always, in the vaccine setting, always thought very carefully about what we would go to the FDA or to a regulator with in terms of a data package, in case then that became sort of hardwired in as a routine. So being clear, the analytics you do to characterize a process, to validate a process, show robustness and consistency, but then the subset of assays that you use on an ongoing basis to support that process and the release of that product. Because if you're not careful, you can hit yourself with a massive burden, not just of cost, but also potential batch failure.
Yeah, so and we saw that with Novartis right at the beginning with Kymriah, right? I mean, a lot of them didn't technically pass because the bars were set so high and so strictly.
I think quality, when I think about it, right, and being a person that did a lot of the audits of our CDMOs and CROs myself in the earlier days, right, quality has to be the center of everything we do, even when we scale up, when we become better at cost of goods, right? Because to me, the essence of quality, right now I know it might mean that few analytical test requirements will go down, right? Just like in biologics or monoclonal antibodies, you don't test every batch for every single process and every step of the way. But until we get that database of the information together, I think it has to continue with the same rigor. Because a lot of these things we're still in the forefront of innovation. It's like, you know, iPhone just came out and we're still in the world of Blackberries. And so we think because we're in the iPhone first user advantage, we're like, oh, we got this, let's move on. But I think the rest of the world and the rest of the diseases and things that we target, we still have to catch up before iPhones become normal. That's kind of my generic analogy. I spent a lot of time with my family talking about what we do, so bear with me on that one. But to me, when I think about advancing things, analytics, the only way we can get there faster is by sharing. So our industry, you know, one of the first lessons I had from Dr. Fovio from Genethon, I think he was there, right? Yeah. 8 years ago, was VCN. I'm like, this company reported this VCN of 8. FDA says 5 is the max. Ours is 1. What do we mean, right? Well, even the simple terminology of VCN, how you actually conduct the test, what the controls are, determines that it's actually not apples to oranges comparison. It's almost like an apples to oranges, but not apples to apples comparison. So I think we need to, as an industry, share more knowledge on what are the tests we're doing, how are we doing it, and actually only way we can succeed is if all of the CDMOs, the companies get together saying, hey, some of these things are not really IP or patent. Let's share the knowledge, let's make it a platform thing. That way, quality, not standards, but standards remain the same, but quality testing requirements could come down. We're not there yet because most biotechs or most players in cell and gene therapy have experience with N of 1 or 2 or 3 compounds or products at most. I think that's the challenge we have.
Wanted to add another angle along the same line. I think when we talk about quality, is not that GMP compliance and GMP readiness. For this industry has a broader term and application. When it comes to quality, we would need to consider also aspects of process robustness, process characterization, the type of methods and assays. And analyticals are becoming the bottleneck to some extent for the field. The type of analytics and then how robust they are, even the characterization methods that we are using, how much data is available to be able to make the right decision. Also in process control, in process monitoring methods, PAT is becoming a major factor how we can make these therapies more effective. Because at the end of the day, when it comes to the future commercial viability, you need to have sufficient data, sufficient understanding of the process, and then, of course, in a phase-appropriate manner, moving from clinical to the commercial, you will need to rely on the data. And having robust analytical methods, broader understanding of the quality of the process and product will become critical.
Will AI sort that out?
That'd be amazing. We have so much data. Every one of us shares it.
Yeah.
It's like a registry. Have AIs do the transnet. That would be great.
Sorry, I wanted to, we talk about AI, and over the last 10 years, I've been working on characterization in particular process and also analytical methods. One aspect that we felt that the field needs significant improvement or opportunities are comprehensive characterization methods. We have been looking at whole genome sequencing, gene array analysis, and others. But yes, we can have very good methods. At the end of the day, it comes down to data interpretation and bioinformatics and understanding the data. And that's where AI and machine learning can help. Because you can have all these methods, but if you cannot make sense of the data, then it will become challenging. And I think that's where AI can help.
Yeah, I think we need more computer scientists in this room, starting very soon, if not right now.
I think there's maybe one other aspect here as well, and that I found myself in a very interesting discussion just yesterday when you think about robustness and variability. I think, to your point, Kinnari, when you make more than one product, you can start to see how changing the gene of interest or that cassette can dramatically affect product quality, even if you hold all other things constant, right? So how do we understand when we're designing a transgene or a promoter or any of the other components that go into a gene therapy, what that's going to mean for robustness and variability? Then on the other side, there's all the parameters that go into the engineering, right? And the problem to me is we don't get that many bites at a cherry here. How many batches do you really make in the life cycle of development program? And the emphasis is fix your process as soon as you humanly can. So you're not going to optimize anything beyond that for fear that you're going to suddenly have a comparability issue and add 2 years to your timeline. So I think we need to get a little smarter about which of these issues are intrinsic and immutable and based on perhaps a suboptimal design of a payload versus which are engineering problems that can be solved by understanding how we make this again and again and again.
But it's still biology, right? So there's so many variables, and I think that's where maybe we will run up against some limitations, because I was talking to Devin Smith yesterday, and he was like, all of a sudden a batch doesn't work, and we don't even know why. And so I think from a data interpretation perspective, that will probably limit things. I don't know, back to synthetic biology, maybe?
Yeah, so I think AI, machine learning, they're loosely used terms. And if I talk to somebody in AI and machine learning, they laugh at me when I use that terminology. So it's a branding name, let's be honest. But the one thing I have learned is the effectiveness of it is totally reliant on as much data as you can get your hands on. And to your point, you don't produce, for gene therapy, you don't produce many batches. Right. To get into the clinic, you're producing scarcely anything. So the only way you're going to get a sufficient body of data is your point of it has to be through collaboration. Yeah. And we have to customers of CDMOs have to sign some sort of a waiver to say, I will allow you to pool my data along with everybody else's and apply algorithms to it, because it will benefit me indirectly, but it will also benefit everybody else. And I've tried to, in the previous life, at one point, I tried to get people to agree to this, and it was really difficult, but it was the only way that you were going to get a sufficient body of data to the odd spurious result. It's analyzable with sufficient data by machine learning techniques. I've seen that applied in other things, but it's going to rely on people agreeing to share.
I think it's going to be not just customers of CDMOs, but CDMOs as well. They have to share, right?
Yeah.
And health authorities. I think all three parties need to come together to have this conversation in an unblinded way of what data we have and what does it mean.
I think Ginkgo are having a little bit of that approach in their work, right? I don't know if people are familiar with that, but Ginkgo Bioworks, they do development work on behalf of customers and on the premise that they can use the data and develop machine learning algorithms, et cetera. So last opportunity for the audience. There's a question over there. Stephen.
Is that box heavy?
I don't know.
No, okay.
Thank you.
Look out.
Hi, everyone. I'm Stephen Ward from the Catapult in the UK. That was great. Really enjoyed the discussion. And I think two comments. Firstly, we still seem to be talking around testing quality in at the end, which is just it's older than me. We've really got to, have this more predictive process sort of technology mindset. And so it's good that people are now really starting to engage with structured data and PAT, et cetera. It's something that we've been trying to do for quite a while at the Catapult. And secondly, I thought it was really refreshing to people talking again around sort of democratization of data and sort of pooling of data, but without losing control of your data. I think that's fantastic. And completely agree with Kinnari's point around this shouldn't just be in CDMOs. This should be open open source. And I think it's not just being talked about. There's some forming workshops going on in the UK over the last 3 or 4 months. Something called a putative sort of digital medicines manufacturing sort of data institute to look to pool data in a certain way. This is from APIs through to ATMPs to aggregate up that data set to give that more critical mass in data, but of course you need the correct format, quality of the data, scrubbing, all of that good stuff. So how can we learn from other sectors who are already doing that really well, rather than us always thinking we have to do something special in ATMPs, because there's a lot of businesses out there that have already done this much better than we have. So I thought it'd be a great session, but I'm just really encouraged that that conversation is coming through. And I think, as I'm actually speaking to someone at lunch around this as well, it's something that I might want to pick up a little bit in the future as well, around how we might create that safer space, if you like, for data aggregation. So thank you.
Thank you, Stephen.
Yeah. No, I think it's good. It's really funny. I mean, it's just suddenly remember. So my first time I was involved in a conversation around standardization in gene therapy was, if anyone remembers, Williamsburg back in the 90s. So it was like late 90s, they tried to come up with reference standards for adenovirus, and they came up with, they had a batch manufactured, they sent it out to all the CROs, everyone ran their analytical methods on it, and they looked for like this, coming up with a standardization titer that could be used in all assays. And that's, I mean, nobody uses adeno, but that whole initiative, we've tried it before. We need to really get engaged in it and seriously push. I know there's more recent initiatives to try and do this, but things like that get everybody on board and actually do. I mean, we're all going to be successful together at the end of the day. There's no point in trying to have sort of petty rivalries. There's going to be, if we're going to get a standardized platform that the FDA is going to accept and get behind, and we're going to be able to do this iterative design without going back to the beginning. The only way that's going to happen is, as an industry, we work together and collaborate, support each other, and get involved in this data sharing, and get involved in setting these reference standards, consistencies, and assays. Otherwise, we're going to hold this industry back. Patients are going to be waiting much longer than they need to be for us to get our act together and get products out there. So, some new fire behind those sorts of initiatives would be good.
I feel like that was a fantastic closing statement. I would like to give the other members the opportunity to do the same.
Maybe I'll go next in a sense that I just wanted to piggyback off of. Dr. Claire Booth from UCL Gosh, right? I'm part of the Agora nonprofit organization that's helping cell and gene therapies become accessible because that have clinical data, but unfortunately the companies did not survive the harsh financial markets. There, we're spending a lot of time looking at how much does it cost to develop a therapy in cell and gene. And the costs are enormous, whether you're treating and making it accessible for 25 patients or 20,000 patients. It's costing north of $150 million for each asset to get to a BLA, right? We don't have that luxury and that kind of financial means to continue every single disease, 8,000 plus diseases at a time in that methodology. So I think the curiosity and collaboration, let's remain curious on how do we do things efficiently better including future technologies of non-viral backbones and et cetera. But let's be collaborative, because none of us can individually figure this out. And by the time we get there, it's not going to work, right? It's going to be too late for these patients.
Lisa?
Yeah, I mean, completely agree. I think it's a great kind of, theme to come out of this session, is we definitely have to work together. And, last year at the beginning of the med meeting, there was a mom who spoke in the keynote. I believe her son had DMD, and his name was Joseph. And she said a statement that I think about every day, and I tell my team they're exhausted by how many times I talk about it internally, because she said, my son Joseph will either be the last, that generation will be the last to die or the first to live, and that's in your hands. And I think about that all the time, because the work we do is extremely hard. We can't do it alone, and we have to constantly think about, the North Star, which is the end of the day, how do we get them into the hands of the patients? And, we really do need to collaborate. We need to be innovative. We need to think about as much ways as possible to be able to achieve that goal. And it is with us, right? And constantly, I think, especially our teams that are coming from academia, there's this quest for perfection. And, we're going to have to, it's got to be fit for purpose. It's got to be 80 to 90% yes, but it's never going to be perfect, right? So how do we go and really focus on quality, but also speed?
Talked about the future of genetic medicine, very important topics, but also we need to talk in the future about the automation. How can we make these therapies commercially viable, and automation, closing the process, reducing the GMP manufacturing risk are key topics out there that we need to talk about them. I just wanted to highlight, but important topics that we discussed today.
Richard?
And what's left to say? I would.
I'm sorry.
I mean, maybe to no. It's I do completely agree. I think collaboration is the way forward. I know we've talked a lot about manufacturing here, but I think about what we saw in the DMD space, with everyone getting together to solve the safety problems by 4 sponsors getting together and sharing their data sets, which is kind of not something that's normally been a thing in our industry, right? So I think that mindset, when the problem's big enough and we see there's a need to do it, I think companies will come together and do the right thing. I'd also like to see how we can optimize taking costs out of running clinical trials. Outside of manufacturing, that's the biggest thing that we're all wrestling with on a day-to-day basis. How can we bring more of that monitoring of patients into the home? How can we use things like wearables? How can we really change the paradigm for how we measure the benefits here? But I'm optimistic. I couldn't be more fully behind where I think we're going with this technology. It's going to happen.
Well, thank you, everyone. Thanks to the audience for your contributions, and we'll see you out there.